this was really a combination of many, many years of work, both when I was at Fred Hutchinson Cancer Research Center and where I'm now at MD Anderson Cancer Center, work done by Jinmei Wang, who's the lead author, and the collaboration of several people who helped us to first demonstrate that IL-21 acts through phospho-STAT3 as one of the primary downstream signals leading to upregulation of CD28 and CD127 in human CD8 T cells. And I think the reason why it really took so long to do this study is obviously there are no transgenic or knockout humans, at least not ones that we can engineer or ethically safe to engineer. But although there are patients, which we do use in this study, who have a STAT3 dominant negative phenotype. These are patients with Job syndrome. And in these patients who have some dysregulation or lack expression of STAT3, turns out we could not generate memory cells with IL-21 priming. So exposure of these T cells in the naive state to IL-21 did not lead to a generation of essential memory population antigen-specific T cells, because STAT3 was one of the important signaling molecules for that to occur. And phospho-STAT3 sort of links the receptor binding to the promoter region of CD28, which we demonstrate by ChIP-seq analysis. That was fundamentally important. But even more striking was the fact that as T cells differentiate into effector cells and terminally differentiate effector cells, that leads to a chromatin condensation or a chromatin pattern that did not allow for phospho-STAT3 binding to this region of the CD28 gene. And so logically, to overcome this differentiation obstacle, I guess one could say that, we would try and use different HDAC inhibitors. And as it turns out, SAHA or Renostat was effective in allowing that part of the CD28 gene to be exposed. Additionally, other HDAC inhibitors were found to be less toxic and at least equally, if not more effective, when used prior to IL-21 treatment, leading then to a generation or enrichment of a central memory phenotype. So I think that's important when one is dealing with a TIL population or a CAR T cell population, where the T cell product is largely effector, even sometimes terminal effector cells, which may have high cytolytic capacity, but certainly not a high replicative capacity. And so we went and actually took those TIL cells or CAR T cell, TCR-T engineered cells, and so that while IL-21 had no effect in generating memory cells from those antigen-specific populations, it was possible to reprogram them using a protocol that involved HDAC inhibitor preconditioning. And I think that's important for the field in a way when you're a little bit worried about things like additional regulatory constraints, the safety issues associated with genetic engineering. Here, you're not permanently altering a genetic code or putting in a transgene. You are sort of using nature's own million-year evolution of the T-cell signaling costimulatory memory pathways and just kind of nudging it a little bit in the direction that you want. And we've proven in a number of studies already published that generation of central memory T cells using a similar workflow can lead to long-lasting in vivo responses and persistence of a transferred T cell population. So we think it's an interesting paper. I'll let other people decide how important it is. And I really do have to commend Jinmei Wang on the diligence and hard work that was put into something that took years and years and years to reach a point where we could unequivocally state that this is a method and a mechanism at play for the generation of central memory T cells for adoptive therapy.